cooling above freezing temperature? The first is cold shock, which is caused by cooling above freezing temperatures. This is more important for ova, not very much important for sperms. This occurs up to 0 degrees centigrade. When the temperature goes from 0 degrees and minus 80 degrees centigrade, the osmotic shock and formation of intracellular ice in ova and sperm takes place. For successful cryopreservation, these things are controlled. In short, the straws containing diluents are transferred into canisters. The canisters holding straws after allowing varying equilibration period are dipped first into liquid nitrogen vapors and then into liquid nitrogen at the temperature minus 196 degrees centigrade. Legendre and Billard stored the sperms of rainbow trout in dry ice or in liquid nitrogen up to 6 months. In a number of freshwater species, freezing has been carried out by pelleting suspend sperms into dry ice. The transition from 0 degrees to minus 70 degrees centigrade lasts approximately 2 minutes, resulting in a rate of 35 degrees centigrade per minute. Since the rate decreases above minus 60 degrees centigrade, the best results are obtained in liquid nitrogen. The meal should be frozen immediately after collection. The freezing should be rapid so that thermal shock is minimal and not so fast as to allow the formation of cellular ice crystals. The seal visitibus required to be cooled at 15 degrees centigrade per minute by programmable cooling chamber, while straw freezing is possible by manual evaporation over lithium N2 in the thermocol chamber. Then how to store? Liquid nitrogen at minus 196 degrees centigrade is the most commonly used cryogen. The freezed milk straws or visitubes are immediately immersed in liquid nitrogen and kept undisturbed. The evaporation loss of liquid nitrogen is compensated by regular filling to the container. To manage cryobanks efficiently, it is essential to keep comprehensive records of all stocks preserved. Next, coming to thawing technique. What is thawing? Cooling and thawing rates are among the most critical variables affecting the success of cryopreservation. In case of carps, spermatozoa fast thawing is preferable as slow thawing can be recrystallize the small intercellular crystals which may damage the cells. The cryo-milled samples of carps can be thawed in warm water of 38 plus 1 degree centigrade for 7 to 9 seconds. Sperm motility should be estimated. Qualitative observations of thawed sperm can be quite helpful in evaluating protocols. For example, if the sperm cells are visibly damaged, the cryoprotectant concentration may have been too low or the cooling rate may have been too rapid. Conversely, if the cell morphology is intact, although the sperm are immotile, the concentration of the cryoprotectant may have been too high. This would be confirmed by a reduction in the equilibration motility. Cryopreservation of eggs and embryos. Unlike spermatozoa, Attempts to cryopreserve fish eggs and embryos have been unsuccessful. During cooling, due to the relatively large size of fish eggs, which contain relatively large volume of yolk and impermeable perivitelline membrane, are challenging problems. However, in recent years, methods for the cryopreservation of the eggs, embryos, and larvae of many invertebrates, such as oysters, mussels, rotifers, and ragworms, have been reported. Among several cryoprotectants used, it has been reported that methanol was the most effective than either DMSO or erythritol for zebrafish. It has been observed that methanol penetrated the entire embryo within 15 minutes, and other cryoprotectants exhibited little or no penetration into yolk, even after 2.5 hours. In zebrafish embryo, the permeability of the methanol appeared to decrease during embryo development at 22 degrees centigrade. The use of ultrasound technique has been reported to enhance the embryo embryo permeability in zebrafish. However, methanol was demonstrated to show a limited degree of penetration into prime 6 stage of zebrafish embryos. But it did not penetrate in the subsequent stages. However, observed that DMSO is a good cryoprotectant for Medaka fish embryos. In common carp, the morule stage embryos are found to be partially protected against chilling in DMSO and sucrose mixture. Half epiboly in DMSO, sucrose and methanol solution, and heartbeat stage in the methanol and glycerol solution. Using isotope labeled DMSO and glycerol, it has been found that these solutes penetrated into both dichlorinated and intact zebrafish embryos after a duration of 5 hours while working on the hatching of common carp. Embryos stored at 4 and minus 2 degrees centigrade in different concentrations of methanol and sucrose found that the hatching performance was at its maximum in sucrose at 4 degrees centigrade. No survival was observed at minus 2 degrees centigrade in any concentration of sucrose solutions. While studying the hatching responses of rohu embryos at different concentrations of cryoprotectants and temperatures, reported that the hatching performance of embryos stored at 4 degrees centigrade temperature in combination of methanol and trehalose showed the highest percentage of hatch out, that is 72 percentage. However, it is further mentioned that cryopreservation of fish, egg, or embryos could not be made successful because of high chilling sensitivity and low membrane permeability. They further suggested that the cryopreservation of an isolated embryonic cell is one of the options to preserve both maternal and paternal genome. Relatively, attempts to cryopreserve invertebrates, especially crustacean egg, embryo, and larvae, have been more successful than the finfish. For example, in peanut shrimp, the successful survival of thawed egg and larvae have been reported up to freezing temperature of minus 10 degrees centigrade. While studying chilling sensitivity of zebrafish oocytes, reported that the oocytes are very sensitive to cooling temperatures and their survival decreased with decreasing temperature and increasing exposure time periods. Further, it is mentioned that stage 3 oocytes are found to be more susceptible to chilling than stage 4 oocytes, and that individual females had a significant influence on oocytes chilling sensitivity. This freeze cooling sensitivity factor may also be one of the hurdles for the development of protocol of cryopreservation technology. Fish embryo cryopreservation no doubt is very difficult due to number of hurdles during cryopreservation process. Therefore, recently more focus has been laid on fish oocyte cryopreservation. The advantage in the oocyte cryopreservation is because of their small size, high membrane permeability, and less chilling sensitivity features. What is the importance of cryopreservation? Asynchrony in spawning state between sexes can often result in a shortfall of gametes of the required quality and quantity. Depending on species and biogeography, male and female fish can mature at different times in a spawning season. Under these asynchronous conditions, especially during the beginning and end of the spawning season, gametes can be collected and held under appropriate storage conditions actual for later use. Similarly, if milk or ova of special interest are required for hybridization, they may need to be stored until fertilization can be carried out. Few male brood stock can be maintained and effective population size can be increased. Cryobanks can play a crucial role in the genetic management and conservation of aquatic resources. Through frozen sperm banks, genes from valuable individuals or stocks which have desirable characteristics, example, for use in inbred lines and selection programs, can be preserved for further use and development. In such case, representative genes in the form of sperm can be selected for cryopreservation. Sperm cryobanks can also be used to manage the genetic integrity of farmed stocks. For fish species, especially those which have a short cycle, such banks are of particular importance to protect against inbreeding, founder effects, and random drift of captive stocks. To minimize these detrimental effects, milk from the representative stocks can be cryopreserved and genetic material periodically reintroduced to increase the heterozygosity in stocks or to increase the contribution of desired genes in the offspring. In selection programs, milk from original stocks can be cryopreserved and used to make half sibling comparisons at a later date. During disease outbreaks, entire stocks can be affected. Therefore, periodic freezing of milk from disease-free males can help to buffer farms against such catastrophes. The use of post-thawed milk to fertilize ova from similar species or strains can aid the rate of recovery of farms. In instances of Scarcity of males. Example, Protoandrous hermaphrodite species of common interest such as groupers. Milt can be stored for later use. The value of cryopreserved milt is even greater in species such as bass, Ephinophyllus malabaricus, which can be take up to 8 to 10 years to mature, which can be taken up to 8 to 10 years to mature. Now, if you look at the summary of this chapter, germplasm cryopreservation includes storage of the sperm, eggs, and embryos, and contributes directly to animal breeding programs. In fish aquaculture, the successful cryopreservation of gametes and embryos could offer new commercial possibilities, allowing the unlimited production of fry and potentially healthier and better condition fish as required. Aquatic species preservation would assist the development, protection, and distribution of research lines and would offer benefits for restoration of endangered species. Cryopreservation is a process where cells or whole tissues are preserved by cooling to low sub-zero temperatures such as 77 Kelvin or minus 176 degrees centigrade, which is the boiling point of liquid nitrogen. Motility is the most commonly used parameter to evaluate sperm quality since sperm must be motile to achieve fertilization. Normally, the sperms are collected from healthy fishes by stripping method. The catheter may be used, which is better alternative to stripping. The sample should be free from urinary and fecal matter. There are two basic extenders. They are called Monib's medium and Manezo medium. The efficiency of cryopreservation is greatly enhanced if the pre-frozen milk is diluted with a suitable extender. The cryoprotectant's main function is to penetrate in the cell and may help spermatozoa to tolerate freezing temperature. Care must be taken while freezing live material since the water it contains expands during ice crystal formation, potentially rupturing the cell. Rupturing the cell should be controlled. Cryopreservation generally means storage of cells or tissue at 196 degrees centigrade, the temperature of liquid nitrogen minus 196 degrees centigrade, which is also called as the temperature of liquid nitrogen. It is the most commonly used cryogen. Cooling and thawing rates are among the most critical variables affecting the success of cryopreservation. Unlike spermatozoa, attempts to cryopreserve fish, eggs, and embryos have been unsuccessful. However, in recent years, methods for the cryopreservation of the eggs, embryos, and larvae of many invertebrates, such as oysters, mussels, rotifers, and ragworms, have been reported. With this, we have come to the end of the chapter, and these are the model questions. Answer the following in about 30 lines in each. Write in detail about cryopreservation. Write about quality evaluation of milt. Write about collection of milt. Write about preparation of extender solution. Write about selection of cryoprotectant. Write about freezing and thawing. Write about cryopreservation of eggs and embryos. Mention the importance of cryopreservation. Thank you.